All students, I am Prasad GC, lecturer in chemistry. Here I am discussing about one of the topic of basic analytical chemistry, 5th semester BSc course, Mysore University. Title of the topic is chromatography. Firstly, we go through lesson objective to learn column chromatographic technique and use it as a tool for separating components of a mixture on laboratory and industrial scale whenever and wherever it is required. Learning outcome: students will be made to understand different types of chromatographic techniques. Introduction: column chromatography. Column chromatography in chemistry is a chromatography method used to isolate a single chemical compound from a mixture. Chromatography is able to separate substances based on differential adsorption of compounds to the adsorbent. Compounds move through the column at different rates, allowing them to be separated into fractions. The technique is widely applicable as many different adsorbents can be used with a wide range of solvents. Adsorbents such as normal phase and reverse phase or otherwise. The main advantage of column chromatography is the relatively low cost and disposability of the stationary phase used in the process. The latter prevents cross contamination and stationary phase degradation due to recycling. Column chromatography can be done using gravity to move the solvent or using compressed gas to push the solvent through the column. Column separation. In column separation, a column is prepared by packing a solid adsorbent into a cylindrical glass or plastic tube. The size will depend on the amount of compound being isolated. The base of the tube contains a filter, either a cotton or glass wool plug or glass frit to hold the solid phase in place. A solvent reservoir may be attached at the top of the column. Two methods are generally used to prepare a column. One is dry method, another one is wet method. For the dry method, the column is first filled with dry stationary phase powder, followed by the addition of mobile phase, which is flushed through the column until it is completely wet, and from this point is never allowed to dry. For the wet method, a slurry is prepared of the eluent with the stationary phase powder and then carefully poured into the column. The top of the silica should be flat and can be protected by a layer of sand. The individual components are retained by the stationary phase differently and separate from each other while they are running at different speeds through the column with the eluent. At the end of the column, they elute one at a time. During the entire chromatography process, the eluent is collected in a series of fractions. Fractions can be collected automatically by means of fraction collectors. In this case, multi-stream collectors are used. The composition of the eluent flow can be monitored and each fraction is analyzed for dissolved compounds. Example by analytical chromatography, UV absorption spectra, or fluorescence. Colored compounds can be seen through the glass wall as moving bands. Stationary phase. The stationary phase or adsorbent in column chromatography is a solid. The most common stationary phase used is silica gel, the next most common being alumina. Cellulose powder has often been used in the past. A wide range of stationary phases are available in order to perform ion exchange chromatography. There is a important ratio between stationary phase weight and the dry weight of the analyte mixture that can be applied onto column. For silica column chromatography, this ratio lies within 20 is to 1 to 100 is to 1. Depending on how close to each other the analyte components are being eluted. The stationary phases are usually finely grinded powders or gels or micropores for an increased surface. There is an important ratio between the stationary phase weight and the dry weight of the analyte mixture that can be applied on the column. For silica column chromatography, this ratio lies within 20 is to 1 to 100 is to 1 depending on how close to each other the analyte components are being eluted. Mobile phase. The mobile phase or eluent is a solvent or a mixture of solvents used to move the compounds through the column. It is chosen so that the retention factor value of the compound of interest is roughly around 0.2 to 0.3 in order to minimize the time and amount of eluent to run the chromatography. The eluent has also been chosen so that the different compounds can be separated effectively. The eluent is optimized in small scale pre-tests, often using thin layer chromatography with the same stationary phase. Keep a piece of glass wool at the bottom of the column and pack it with a suitable stationary phase like silica gel as compact as possible. Pour the mobile phase from the top of the column and open the tap to wet the stationary phase completely. Solvent level should be little above the level of stationary phase. Carefully place the sample mixture to be separated on top of the column. That means preferably with the same solvent as of mobile phase. Open the tap and allow the mixture to percolate through the column. Start addition the eluent slowly from the top in small quantities, keeping the column wet always. As the eluent moves down, different components of the mixture move down the column at different rates based on differential adsorption. The component that moves faster leaves the column first, followed by the components in order and are collected separately. The technique. Key steps in the ion exchange chromatography are listed below. First one, an impure protein sample is loaded into the ion exchange chromatography column at a particular pH. Charged proteins will bind the oppositely charged functional groups in the resin. A salt gradient is used to elute separated proteins. At low salt concentrations, proteins having few charged groups are eluted, and at higher salt concentrations, proteins with several charged groups are eluted. This is about some of the techniques of chromatography. You may go through reference books. Those are all Fundamentals of Analytical Chemistry by Skoog and West, Principle and Practice of Analytical Chemistry by Fifield and Keeley, Qualitative Inorganic Analysis by Vogel, Analytical Chemistry by Treadwell, etc. These are some of the reference books regarding this topic. Thank you.